So if you were interested in trying to understand at the DNA level what kind of glitch might have caused a particular consequence, a place to start might be the exons. They're the part that code for protein. If you were looking, to go to the place where you're most likely to find a significant result, that would be a good start. And it would save you a lot of work, because the exons, it turns out, are only a minor part of the entire genome. Again, exons, introns, in between them are making up a gene, and exons only make up about 2% of the whole human genome. In fact, we call all of the exons, just because we like to make up new terms, the exome. So you have the genome, which is all of the DNA, the exome, which is just the protein coding parts. And if you had a way to take a DNA sample and just look at the sequence of the exome, you'd have only 1/50th the amount of work to do. And then we always like that kind of scene when it happens. Well, it turns out we do have a way. It's a very clever way to be able to pluck out of a particular DNA sample just the exons and just look at their DNA sequence and see if you find something interesting. So this is called exome sequencing, and it has advanced the field of medical genetics substantially in just the last few years. Let me tell you a story where whole exome sequencing led to a pretty dramatic outcome. This is about Nick. That's Nick's picture there, now at about the age of 6.5, and actually doing very well now, but he was not doing well when he was first brought into medical attention, because after what seemed to be a fairly normal first year, he developed severe inflammatory bowel disease, something called Crohn's disease, which usually doesn't appear in such a small child. He had 100 surgeries, more than 100 surgeries. Every time he tried to eat solid food, the disease would get worse, so he was being fed intravenously. Nobody knew what was wrong with Nick, and he was wasting away. His doctors, at their wits' end, decided, well, let's just try to get more information. Maybe we'll sequence his exome. This was a group in Wisconsin. And they found a mutation, a misspelling of a gene called XIAP, which nobody had paid much attention to before. And it had been loosely linked to a blood disorder. And in the rare patients who had that blood disorder, bone marrow transplantation had actually been successful. But those patients didn't have the intestinal problem that Nick did. As Nick was getting weaker and weaker, they talked with his parents and decided, with a full ethical board review, that the best chance for Nick was a stem cell transplant. With no confidence this was going to work, but the DNA results suggested it was maybe the best hope they had. So he got a stem cell transplant 2 years ago, and I'm happy to tell you, Nick was in regular school this past year. Nick has a hamburger whenever he wants to, and that all goes well. And this is essentially now a normal little boy. And had it not been for this information that came from exome sequencing, I doubt if Nick would have survived. That's a good story. I'll tell you another good story. Noah and Alexis Beery, these are twins, boy and girl, who early on in childhood both developed neurological problems that appeared to be some form of dystonia, a difficulty in terms of the tone of the muscles. They went to numerous neurologists. Nobody could quite figure out what this was. Because they were twins, not identical twins, but fraternal twins, and they were both affected, this sounds like a recessive disorder, and the assumption was it probably is, but it didn't fit anything that had ever been described. They were tried on L-DOPA, the same drug that's used for Parkinson's disease, and they got some benefit from that, but it gradually became less and less valuable and successful. And Alexis particularly got into more and more trouble with falling. This is a video, it's a little hard to watch, of her instability and her mom taking this picture. And with more and more falling, more and more difficulty actually with breathing, many times in the hospital, emergency room visits, parents at their wits' end. Noah also affected, not quite as severely. So the parents heard about the idea that maybe DNA analysis could help and brought both their kids to the Baylor College of Medicine in Texas. And after much evaluation, the group decided, let's do the exome sequence and see what they can find. So they read out all the ACs, Gs, and Ts, and they found something that had never been described before. basically a glitch in both copies of an enzyme gene that normally is responsible for making a substance that you need for neurotransmitters to work in the brain properly. And the beauty of this was that knowing that immediately suggested that if these kids were treated not only with L-DOPA, but with another simple compound called 5-hydroxytryptophan, that should help compensate for their problems. So they were started on that. Within a few weeks, they seem to be looking a lot better. I've met the parents. The parents sent me a video of Noah and Alexis taken just a couple of months ago. Now they've been on the therapy for about a year. And this is Noah, and that's Alexis. That's the kid who was spending most of her time in the hospital. She's now running varsity track in her high school, I kid you not. I wish I had more stories like that. These are still few and far between, but it does give you a sense of excitement about how this new window into the molecular basis of illness can give us answers that don't have to take 40 years to result in advances. At least some of the time, you can get something very quickly. Well, again, we'd like to see more of those examples, and we have lots and lots of needs to do so. If you look at what's happened over the course of the last 25 years in terms of the discovery of the molecular cause of illnesses, it is pretty dramatic. Going back to the 1980s, where we really didn't know on the basis of DNA misspellings what was the cause of most of these conditions. Now we're up to 4,700 of them where we have answers. And that is going up even more dramatically now that we can do this whole exome sequencing, because a lot of things that were out of reach are now revealing their secrets. So where does Moebius syndrome fit? Well, first of all, I'm not an expert in this disorder. I am trained as a medical geneticist. I have seen occasional individuals with Moebius syndrome as part of my clinical activities over the last 30 years. But there are people here at this meeting who are much more expert about the neurological problems, about the developmental issues. But I think what we all can say is that something happens in the course of development, particularly of these two cranial nerves that innervate the face and the eye, so-called cranial nerves 6 and 7. And that in general, this is something that happens during development, that is, during the pregnancy. Sometimes other clinical features are also present. And again, at a meeting like this, with many of you going to be examined by some of the experts will notice differences between individuals, even though there's lots in common as well. What might be the cause? Could this be a vascular problem where there is some temporary blood flow that gets diminished just at a critical moment where these cranial nerves are trying to form in that developing individual? Could it be some environmental insult that we haven't yet identified, that at some critical moment in pregnancy there's a great vulnerability to some exposure to something that we don't realize is actually not safe? Or are there genetic factors involved? And it doesn't necessarily have to be one of these. It could be a mix of all of these. In fact, oftentimes what we learn as we begin to investigate is that conditions have a mix of genetic and environmental factors, and sometimes others as well. So why are we talking about genetics at all for Moebius syndrome? Well, in part because there are clearly families where the disorder recurs. I mentioned there are rare families where it's inherited in a dominant fashion, being passed from parent to child. There are other rare families where it appears to be recessive, where you have multiple siblings affected, but the parents are not. Frankly, though, most of the time, Moebius syndrome seems to come out of nowhere, affects an individual. The siblings are not affected, and we don't understand what's that about. Could that have been purely environmental, or is it a genetic combination? We need to get answers to that. And at the present time, we have, so far, only very sketchy ideas of what's going on. Now, yesterday, at the research meeting, two groups talked about specific genes that they had identified in individuals who had sort of unusual types of Moebius syndrome, which were not actually typical, but involved genes in particular ways that maybe are clues of a pretty important sort to where we're going. So we're just beginning to see the leading edge of these discoveries, but they don't, at the moment, explain most cases of Moebius syndrome. Not there yet. But I would predict that in the next, say, 3 or 4 years, we're going to figure out a lot more about what the role is of DNA changes in this condition. So what genetics could tell us will depend upon what inheritance patterns are found, and I've just mentioned that. Finding what genes are involved, that's a big part of the task. Are there interactions between genes and environment? And this isn't just curiosity. If genes can be identified, does it matter? How can that information be used to do a better job of diagnosing? Would it actually be possible, once you had a specific genetic diagnosis, to make a prediction about which kinds of interventions are likely to work and which kinds wouldn't work? Because you'd like to be able to offer people that kind of information as they're contemplating various options of what they might want to do. So the team that is working with me at NIH and has been involved in this now since that dinner next to Vicki McCarroll 5 years ago, here you see 5 folks who are in my research laboratory on the NIH campus. These are all folks who work in the lab. They are both molecular biologists, and 2 of them are experts in computational analysis, because you can imagine this data is really challenging to sift through, so you need computer experts, Peter and Nari Su. And we also are working closely with a clinical team, the folks that you see here, Carol, Eva, Brian, Irini, and Camilla, who are part of our effort when patients come to the NIH Clinical Center to try to be doing a very thorough analysis of everything we can learn about the way in which Moebius syndrome has affected them and their families. Particularly want to point out Lori Bonnycastle, who's here at the meeting, right over there. Lori has been, and she's a staff scientist in my lab, has been the lead in this enterprise, and will be here for the entire meeting, was here yesterday as well, meeting many of you, and certainly very interested in hearing any ideas you have about your families and anything that we should know as we go into this detective work of trying to understand the causes. And Irini Manoli, who is the lead clinician, who's currently in Greece right now, or she would be here, very much dedicated to trying to help identify any kinds of clues that would be useful in making sense of this complicated condition. But we've relatively a small investment in this compared to 2 other groups that we are happy to say we've had the chance to start collaborating with. Elizabeth Engel, who's not able to be here because of a family illness, and David Hunter, her collaborator, who is here, over here from Boston Children's, and who has come here with a significant team of experts from Boston Children's, have been working very hard to try to characterize some of the neurological differences that happen in individuals with Moebius syndrome, and to do some of the molecular analysis. And we are delighted to have a chance to share information with them. And Ethelyn Wang Jabs, known to most of us as Mimi, and Bryn Webb, her clinical colleague from Mount Sinai, who've been involved in Moebius syndrome for a long time, are also wonderful collaborators and have helped us to identify a few special families that we have brought to NIH for more detailed analysis to try to get answers. So I think it's really a good thing that all of these researchers know each other, enjoy working together. That's the best possible circumstance to try to get answers. So the work we've done at NIH has been pretty limited so far. We've seen 4 families at the NIH Clinical Center, which is the largest research hospital in the world. The NIH Clinical Center, 240 beds for inpatients and lots of outpatient capabilities. Patients and family members have come to our clinical center for 3 to 5 days for a very thorough analysis involving many things that you see here: medical workup, consults with various experts, brain MRI scans, studies of muscle functions, CT scans, photography, blood samples, and a skin biopsy. All of the care is free because this is a research hospital supported by you all, by the taxpayers. The DNA is prepared from the blood sample, and then the exome sequencing is done with my lab, Lori especially, preparing the DNA, and then the sequencing center at NIH actually doing the determination of the sequence, and then this vast amount of data comes and the lab digs through it trying to see what's there.